Our last speaker tonight is Dr. Scott Seville, and he's a rather different kind of a character. He's another one of the faculty at UW Casper, but rather than a new hire, he's been there since 1995, having finished his PhD in 1992 in Laramie, actually, at the University of Wyoming. His area of interest is biology, zoology. That's what the official title of his PhD is, but he's really interested in microbiology. Once arriving at Casper, he quickly became the go-to person in the biological sciences, and he taught a broad range of courses from intro to some higher level stuff. One of his loves and his interest is to bring actual hands-on research to students, to undergraduates. And when I say research, I don't mean like, oh, he threw in a couple of labs for his biology course and everybody got bored out of their skin while they were having to try and make the lab work and then write it up when it didn't work usually. And he actually wants people to think about research. Okay, what's the question you're interested in? How do you form a hypothesis? What's the method for answering that? And then carrying out the method, what results, and so on. And here's where it sort of gets interesting because Scott, as I said, is a microbiologist. And he did this best, or he does this best with his own area of interest. And let me just read you a couple of the titles of his articles, the published articles. Here's a couple. 2 new species of Coccidia from leaf-tailed geckos. A new Coccidian from a Midland brown snake. A new species from the eastern hognose snake. A new species of Coccidia from the Cordilleria striped shrew rat. The Coccidia, of course, as you know, I like the the geckos, the rats, the snakes, etc. But the Coccidia is actually a little protozoa that you measure in microns rather than in anything larger than that. And I discovered talking to him that the Coccidia is one of the largest. Would you call it a genus?
Family.
Families. It's one of the largest families of life in the biological world. And we only know about 10, maybe 15% of them. And they all exist in the stomach of animals. So when Scott introduces his students, it's not to a disease, it's to research and how to find this stuff. When he's not teaching, when he's not researching, Scott has went over to the dark side, became an administrator. In 2005, he became an associate dean of the outreach school based in UW Casper. And from that position, he was wildly influential on how we do distance education here in Wyoming. He helped establish and maintain our outreach offices around the state at like the one we have here at Gillette College, like the one we have at Sheridan College. He worked with the various staff there. He has worked with various aspects of delivery in the outreach school all over the state, even coming down to Laramie, coming down to Cheyenne, and doing things down there. In 2004, he took off in another direction. He became the director of the NIH Wyoming Idea Network for Biomedical Research Excellence, INBRE for in short, which is a multi-million-dollar grant that benefits both students and faculty around Wyoming. And Scott may be one of the best known biologists in Wyoming, not because he's some great researcher and has had his face all over TV, but because he has helped every biology teacher in Wyoming's community colleges, help them do research with the students, help them supply money for everything from specimens to research equipment and so on. So Scott has had an enormous impact on how science, and particularly biological science, is taught throughout Wyoming. And with that, I'm going to let him talk about diseases and should we worry. Dr. Seville, thank you.
Thank you, Paul.
Sure.
All right. Well, what I'd like to do tonight, but I have to admit, or confess rather, that Paul really challenged me several weeks ago, and he has cost me a lot of sleep. Because when I told him, I could talk about coccidia, and he said, Saturday we really like to have something a little more interesting. And I said, okay, so we kicked around a couple of ideas, and one of the things I've been doing for the last couple of years, in addition to my standard courses and the research I do, is I've been teaching a course in global public health. And so we got this idea about emerging diseases, which is kind of a fascination of mine, and I'll show you how it ties back into the basic biology work I do. But he said, yeah, something on emerging diseases would be really good. And then we talked more, and it was about, what's it like here? I mean, what do we have to fear? And so I have been tussling with this topic for weeks. And I got to tell you, I finished the last slide in the hotel room before I came over here, and I kind of had the whole idea together about 4 a.m. this morning when I woke up and said, this isn't working. So we'll see if it works. Okay, so thank you, Paul. I love challenges. If I could just get some sleep. Okay, I want to elaborate a little more on what Paul told you about me and what I do. So what I am is I'm a protozoologist. So I study protozoa. These are single-celled organisms. And I study a particular group that are only parasites. They only live in other things. And so what I study about these things is taxonomy and systematics. So this is how we name them, and the structure in which they are organized, which reflects their history and ancestry, what we call a systematic or a phylogeny. And so the basic questions I ask about this group of, we call them coccidia in the generic sense, and those of you that might have grown up on a ranch might know spring scours and cattle. Oftentimes it's caused by coccidia. If you raise chickens, a lot of people treat their chickens regularly for coccidia because it can really negatively impact egg production and their ability to put on weight. So there are lots of coccidia stats out there, but I don't work in that realm. I'm interested in the biodiversity of these organisms. What's out there? What are they in? Where are they found? And so from this, we're trying to figure out, we're trying to kind of develop a baseline. And Paul mentioned, we know about 15% of all the coccidia in the world. So there's a lot to be to see. Students looking for some discovery out there, this is a great area to work in. And we want to find out what's out there, find out how these things are related using molecular approaches, so developing family trees. So we can explore the past, we can understand the present, and we can predict the future. And of course, the future that we'd like to be able to predict is when might these things cause an outbreak? When might they become epidemic? When might a particular population of organisms be susceptible to disease outbreaks? And in the realm I work with, frankly, I'm going to talk a bit about humans, but most of my work is with wildlife. And Paul mentioned a number of things that we've worked with. Unfortunately, those are really neat names. That gecko is from Madagascar. The striped cordillera shrew is from the Philippines. And I still haven't figured out how I get to go in the field and collect these things. I just get the poop. And so I have friends that get these grants to travel the world studying mammals and reptiles and birds and documenting biodiversity. And they always say, send me a box of vials and I'll send you back samples. So I sit in my lab in Casper, dream about the Philippines, and look at poop from striped shrews in the Philippines. Okay, As Paul mentioned, I also teach courses, and in my research, I work with a lot of undergraduate students and encourage and support a lot of other people to do the same, okay? And so I just want to mention, and I'll try and tie this back in because we're all in Wyoming. It's interesting to hear what's going on around the state and what kinds of opportunities are out there for our residents. I'll try and drag in some of the work that people are doing around the state that are supported through our NIH grant, this thing called an inbred, which means I get endless emails asking me about inbred, and I have to say, no, 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 drop the D, it's inbred. So I'll try and tie that back in. So one of the big projects I'm just wrapping up is, as I mentioned, we're collecting baseline data. We're trying to document all that we know out there. So this is actually a huge library project. We're just in the final stages of publishing what turned out to be a monster of a book. And this is the biology and identification of the coccidia of the carnivores of the world. So we have tracked down the literature going back into the mid-1800s, everything in every language that has ever been written about these parasites in carnivores. So things like bears, lions and tigers and bears, oh my, but all kinds of things I hadn't really heard of until we started looking at things in very strange families of carnivores, and that work has been a lot of fun. But the idea is for somebody out there in practice as a conservation biologist, as a veterinarian, as a resource manager, should they see disease break out in some animals that they are responsible for, this is the reference that they can go to and thumb through this thing and get an idea of maybe what it is, or maybe this is something new, which raises a whole nother bunch of concerns. So what I'd like to do tonight, I'm not just going to focus on coccidia because frankly a lot of them don't really cause a lot of disease. I'm going to focus on some other organisms that are a little more exciting and maybe you've heard a little bit about. And then I'm going to talk about some global things, and then we'll move back to Wyoming and talk about things in Wyoming that are interesting from a disease perspective, and what might be the possibility, probability, likelihood of these things or other things coming to our state and threatening the health of our residents. And what's out there to prevent these things from happening, okay? Does anybody have a better idea? That's what I want to talk about. Okay, but before I start though, I'm looking out at these faces. How many of you are in biology right now with Mr. Craig there? Okay. Okay, so you're getting points for this, right? All right. Good job, Matt. I always hate it when there's like two people, which is what usually happens. Okay, so first of all, let's define what are emerging infectious diseases, okay? So emerging infectious diseases are diseases that have newly appeared in a population that are rapidly increasing in incidence or geographic range. And this is a widely accepted definition with the National Institutes for Health, the World Health Organization, those major entities concerned with such things. And so when we think about emerging infectious diseases, here's a couple you're probably aware of and have heard of. We've got things like Ebola, Zika, human immunodeficiency virus, tuberculosis, Hantavirus, and something called bovine spongiform encephalopathy. It's a real tongue twister. So these are things that in fairly recent history have been in the news, have had people feeling threatened, and I would just mention have some things that are related to them here, if not the very same entities, and we'll talk about some of these. And of course, something I think everybody is very interested in right now, and I got to tell you, every time I walk by a restroom, I go in and scrub my hands with soap, because I don't want the flu this year, but influenza virus. And I think influenza virus is really interesting because you can almost think about it as an emerging disease on an annual basis, because every year that influenza virus is really a unique influenza entity, and the characteristics it has and its ability to jump from human to human and to move across the globe and to cause varying levels of disease in individuals is different. And unfortunately, this year it's a little more severe than last year and for the last several years. Okay, the other, at our international level, just a few years ago, there was an outbreak that got in all the news, and this was the 2014 Ebola outbreak in Africa. And this, I think, got a lot of press. People became aware, in particular, acutely aware of how a disease can cause very, very tragic symptoms, very high level of mortality. You can see from that graphic there, people infected with Ebola, almost 50% of them died. So this was a very serious outbreak, and also that it can spread, and that it can also spread quickly in the world in which we live. And I can tell you, I was assigned to the Ebola task force in Casper, Wyoming in 2014. Over the holidays, we had students, Native Africans that went home, and the question was, do we let them come back? And when they come back, how do we support them? And so there was a lot of discussion, and the city was involved, primary health care was involved, and it was quickly determined that things were being brought under control and those students should be able to come back, and things worked out just fine. And of course, historically, there are some epidemic outbreaks, emerging diseases. We don't think of them looking back like that, but of course, the Black Death or the plagues of Europe and Asia going back to the 13th century. These are classic cases of emerging diseases by the way we define it today. In the 1300s, about half of the population of Europe, it's estimated, died from plague. Plague, we now know, of course, back then it wasn't apparent, but it's actually a disease of rats. It's transmitted by fleas. And when it's in rats and fleas, it's okay. The problem is when those rats cohabitate with humans and the fleas, looking for a warm blood meal, go and bite on the next nearest warm-blooded thing, and it's a person, and they inoculate them with the plague. One of the things that's fascinating about Yersinia pestis and the fleas that transmit it is Yersinia pestis in one particular flea, which happened to be the predominant flea on rats throughout Central Asia and Europe, the oriental rat flea. Yersinia pestis, when it infects a flea, it actually lodges in the esophagus, and it multiplies and it blocks the throat. And so the flea, in trying to feed, well, its throat is blocked. And so it can't feed, so it's starving. So it is biting frantically. And of course, every time it bites, it takes in a little body fluid, which hits that Yersinia pestis clog, which then washes it back out into the wound. And so a perfect mechanism for transmitting the Yersinia pestis from the flea back into the host that's being bitten. We have plague in Wyoming. We talk a little bit about that. It's in prairie dogs. We don't have the right fleas. So the plague in prairie dogs is not highly transmittable. It comes through prairie dog towns. If you're ever out in a prairie dog town and it's deathly silent, and you know that nobody has poisoned those prairie dogs, I'd suggest you go get back in your car and leave that site, because it's probably just had a plague outbreak run through, because it will completely kill every prairie dog. I've had that experience, and it's really eerie. So I think historically a more recent major event you might be familiar with is the 1918 Spanish flu. This was a pandemic of the H1N1 flu virus. Nobody quite knows where it initially broke out. You can see the photograph there from Fort Riley, Kansas. Some people investigating the history of this particular outbreak believe it actually broke out in Kansas. Others have a location in central France. Now the thing with the flu And we're not going to talk a lot about flu. But the thing with the flu is, it is very highly transmittable because it is aerosolized. And it's a respiratory virus. And it goes directly from person to person. When you think back to the plague, and I mentioned the biting of the fleas, well, when the plague really got bad, there are different types of plague. Initially, it's bubonic plague, and you get bitten by a flea, and the bacteria infects your body, and people get these, they're almost like boils, only they called them buboes. So they would get these very localized swellings on the body, and that was bubonic plague. Well, over time, the plague, if it's untreated, can colonize the lungs, and that's when it becomes respiratory transmitted, so aerosol transmitted. And it's at that point when people can now cough on one another, like a few of you have done in this room tonight. That's when it really starts to move and a lot of people get impacted. So with the flu, that's one of the big issues, is it's aerosol transmitted. Which makes it very easy to move from person to person, and we'll allude to that a little bit more. So when we talk about emerging diseases, an important question for epidemiologists, people that study disease, parasitologists, is where are these things emerging from? And based on a lot of research, when you have emerging diseases that become epidemic or serious, it often comes from what's called a host switching event. And so this is when the infectious agent is existing in some organism out there, what we call a reservoir. And oftentimes in the reservoir, it's not causing severe disease, okay? It's not causing severe disease, but for some reason, maybe a mutation, maybe because they're in such continual contact, for some reason, that infectious agent is able to jump from that reservoir host into a novel host, and in our discussion, a human being. So it is switched hosts from the reservoir host, and if it's an animal in the wild or an animal, we call it a zoonosis, and it jumps into a human, okay? It's almost like an invasive species moving on to a new continent, all right? So there's a lot of interesting work on invasive species ecology, and of course, when something gets introduced onto a new continent, well, it just escaped all its predators, it just escaped all its competitors, and if the habitat and there's things to eat and water to drink and mates to mate with, yeehaw! Nothing eating me, nobody fighting with me over my preferred foods, and away you go. Well, if you can host switch as an infectious agent, well, you've just escaped the bounds of the things that your reservoir species has been doing to contain you and minimize disease, and you're able to wreak havoc on the new environment that you've colonized. So this is just an illustration. Some friends of mine at the University of Utah, Dale Clayton and Sarah Bush, have done some really, really fun work with parasite host switching, and they work with lice, which is really nice because lice aren't that bad and they don't give you disease. But they've done some really cute stuff. But I love this, these two phylograms, we call them, one for the parasite and one for the host. Very simple. You can see where the lice on bird D has jumped, or the parasite has jumped from host B down to D. So that's illustrative of a host switch. Now, to bring us back home, a little better example in the human context is the HIV virus. And so on this phylogram, or phylogeny, you can see all those red dots represent places where an immunodeficiency virus has jumped from a simian, a monkey host, to a human. And you can see that this has happened multiple times. This is not just one host switch, one colonization. And you can actually see this in the diversity of variants of HIV that exist in human populations around the world. So as I said, there's a lot of evidence that this happens quite a bit, okay? And the kinds of conditions that facilitate this kind of host switching are the kinds of conditions that we seem to be increasing as humans populate the planet and they spread out and they develop different areas. They're increasing contact, or I'm one of you guys too. We are increasing our contact with different types of different types of animals that could be reservoirs for diseases that could colonize humans. Okay? And we're also dispersing these pathogens via dispersing their hosts. So we're really fond of getting exotic pets. Well, exotic pets brought squirrel pox from the UK to the United States, where it started infecting prairie dogs, and also had was able to infect humans, but not with much effect. So we also move around these hosts and pathogens by accident or by design. So I just have a little diagram there. Sorry for my stick man. I'm not very good with drawing things with PowerPoint. But this just shows a human, and these are some common reservoirs for diseases that afflict humankind. And so here we have simians, like I mentioned, with HIV. We also have Marburg virus, some others that periodically outbreak, in particular in areas where primates, monkeys, and humans overlap in the areas they live in. Birds are another good source. We've got rodents and we've got bats. So as we go on, I'll bring up some examples from these different groups of diseases that are coming out of these reservoir hosts. Now the other thing that can get involved in this that is important to think about is how do these things get from these reservoirs to humans? So I mentioned with the plague, it is the bite of fleas that gets into humans. The disease progresses to pneumonic plague, and then it's spread aerosolly between humans. Oftentimes there's some sort of intermediary. So you can see my little mosquito there buzzing around. And as you think about diseases where mosquitoes are transmitting things from rodents or birds or bats or primates, one we'll talk about in a moment is West Nile virus. Okay? And here's just a list. For birds, we've got West Nile virus. Influenza is important. Psittacoses. I don't know if you ever look carefully in the local airport, but there's signs in every airport about trying to smuggle birds back into the United States, because this is a disease which can afflict humans, and it's pretty common in tropical birds. And so they don't want you bringing birds back in your luggage. Salmonella comes from birds. In bats, we've got Ebola, Nipah virus, Hendra, Marburg, SARS, Middle East respiratory syndrome, rabies, of course, which has a long history. In rodents, we've got Hantavirus, rat bite fever, Lyme disease. Okay, so one of the consequences when you have a host switch like this, and I described this a little bit, is that this new host species has no prior exposure and thus no history of interacting with that infectious agent. So thus they have no resistance, no immunity. So typically these new infectious agents, if they are able to establish, they're highly virulent. Virulent is a measure of how sick they make the individual. Now, over time and over continued exposure, and if that host switch results in that infectious agent becoming kind of resident, if you will, or endemic in that new host species, there's kind of this little tit-for-tat battle that goes on over evolutionary time. And the virulence tends to modulate. It was really interesting when I was getting ready for this talk, I was reading an article about emerging diseases, and here a very, very smart scientist said, yeah, and over time, these things evolve to where the battle becomes a stalemate and the infectious agent essentially becomes inert. And I'm sorry, that's not true. What happens is there's this kind of evolutionary battle between the host and the infection, or the parasite, and eventually end up at kind of a compromise. Because from the parasite's perspective, it doesn't want to kill the host until it's gotten everything it needs and it's reproduced enough to colonize the next host. If you kill the host, you die with the host. So that host has to live long enough for the infection, the parasite, to persist to get to the next host, for the person to be active to transmit to the next host. So there's kind of this balancing act between transmission and how sick you make something in terms of the parasite's development over time. I think a really good example of this is if you think back to Ebola. One of the things for Ebola when it colonized humans, and it came from primates, is it makes people so sick, they are identified so quickly, they can be isolated and handled so carefully that we can cut off transmission. And that's what we did. Now, if Ebola had a delayed incubation period where the person is infectious, but they're feeling okay and they can get on that plane and fly to London and walk through the airport and they're spewing virus as they move through, and then they get sick at some later point, well, now they've just distributed that virus all along their merry way, okay? And I think another good example is to think of HIV, where there's that long period of infection before the person starts to feel sick. And they're fine. They don't know they're sick, but they're transmitting virus. So this idea of virulence and transmission is an important consideration when you think about emerging diseases and their long-term consequences. Now, there are a couple of important epidemiological questions to think about as well. Ecological questions, if you will. So what is the source or the reservoir, which we've talked a bit about? And so here we've got something that comes out of mice. And sometimes mice are really cute. So we've got a cute mouse here. How does that infectious agent get from the source to the human? So if the mouse and the human actually have to come into direct contact, well, okay, it's transmitting the infection. But every human's a dead end. And so the ability of it to cause an epidemic and a serious emerging disease issue is somewhat limited. If it's able to transmit to the human, but the human's able to carry the infection and then transmit it on, and now you got a bigger problem. And things are going to happen much more quickly. So the source or the reservoir of the infection, how is the infectious agent transmitted from the source to the humans, and then how is the infection spread from humans to humans are important things to think about when you think about emerging diseases. Okay, a couple of examples. I'm going to go international first, things with cool names and interesting biologies. Nipah virus. How many have heard of Nipah virus? I see one hand. The ornithologist in the audience. Yes? You've heard of Nipah. Just one person, okay. Nipah is a paramyxovirus. It was first identified in 1999 in an outbreak of encephalitis, so inflammation of the nervous system, if you will, and respiratory illness in Malaysia and Singapore, okay? So this disease, there's an incubation period of about 2 weeks. The illness presents itself, there's fever, headache, followed by drowsiness, disorientation, kind of like a very severe flu, okay? But if it progresses to severe encephalitis, about 40% of people with serious neurological symptoms died, okay? And it also, as many things that get into the nervous system do, it has long-term consequences. That feeling of being tired, muscle soreness, headaches, disorientation can persist for long periods of time. In fact, people from this outbreak are still experiencing some of the long-term effects, okay? So the epidemiologists swept in. There was no person-to-person transmission. But what they found is the people that were getting it were associated with pig farms in Malaysia and Singapore. And so they went to the pig farms and they looked around and they said, okay, pigs have got it. Where are pigs getting it? What other organisms around here? And so they looked up in the trees and they found that in the trees above the big pig farms in this part of Southeast Asia, these are roosting sites for flying foxes, these great big fruit-eating bats. And those bats would go up in the trees, they would defecate, urinate, spit. I don't know what they were doing to those poor hogs underneath them. But in their droppings, in their body excreta, there was virus and the pigs were picking it up. So the humans were infected via pigs that were infected by fruit bats. Okay? In 2006, there was another outbreak in Bangladesh. This was different. In Bangladesh, they don't farm pigs. And in Bangladesh, people started transmitting it from one to another, so it was being directly transmitted. But where were the people getting it? Okay? It turns out that in Bangladesh, people love palm syrup. And there are palm syrup harvesters, much like our maple syrup harvesters. And they climb up in the palm trees and they make these gashes and they put these little gourds up there, these little pots that catch the syrup as it comes out. And they take it to the market and they sell it to people, just direct out of the little pots. Well, it turned out that these collectors were people that were very highly likely to get the disease. So they started suspecting something about those pots. And what they found out was bats love palm syrup too. And they were coming down, upside down on the stalks of the palm trees, sticking their heads into the pots. Well, they were salivating, so there was saliva, as well as when they urinate and defecate from above, it was going into the pots. And so the reaction was, okay, we have to homogenize, we have to treat all of our palm syrup. It needs to be heated to kill this virus off, and that cut off the disease. Okay, here's one that is global, but also becoming more of a concern in North America and maybe someday a concern for us. Okay, this is the Zika virus. Okay. Zika was originally identified back in 1947 in Uganda in monkeys. And in 1952, the first case in humans was documented again in Uganda. It spread across Africa, the Americas, Asia, and the Pacific. And the first outbreak, the first disease outbreak, so it was being observed periodically. Somebody would be diagnosed with Zika. But the first true outbreak was on the Micronesian island of Yap in 2007. Okay. 2015, it appeared in South America, and it very quickly spread across the continent. And also in 2015, scientists, epidemiologists, medical practitioners also recognized that there was a relationship between women infected with Zika and some very unfortunate birth defects, in particular microcephaly. And this is a pretty shocking type of affliction with long-term consequences. And it turned out that there's been a lot of work now that has confirmed that Zika does indeed have this effect on the developing fetus. They also determined that the vectors were Aedes aegypti and Aedes albopictus. These are mosquitoes, and there's a lovely picture of an engorged mosquito there, female mosquito. Okay. And so you can see here, this map just shows the distribution of the disease. So in 2015, I could use my pointer, I guess, and help you out. So in 2015, here was where it was first identified, 16 over here, and it has just spread its way up South America, through Central America, and up into Mexico and the southern United States. Okay. Now, a couple of notes about Zika. Most people that are infected don't even know it. They might have a mild fever, 1 or 2 days of feeling a little under the weather. Okay, so fever, rash, headache, joint pain, muscle pain. spread by mosquitoes, okay? As I just mentioned, it could be passed from pregnant women to the fetus, so what we call congenital transmission, okay? It can also be passed through sex. And in fact, the first case of sexual transmission was documented back in the 50s in Africa in an American medical worker who returned home to the United States and passed Zika to his partner. Now let's look at North America. This shows the distribution of Aedes aegypti in North America. And as you can see, it's pretty widely spread across the southern part of the country. Another feature of this map is it also shows the numbers of people coming from South America that annually travel to those different locations. So individuals that travel to South America have the potential to bring up Zika and bring it into some of those areas where the virus has not yet been observed. And if this works, I'm going to give this a little shot. So look at this. So what's limiting the distribution of these mosquitoes that are the vectors for Zika that affect where Zika occurs is temperature, right? So Wyoming looks pretty good here. However, it's just something to think about off into the future. I want to just show you what temperature looks like over the next 100 years. I'm going to have to. Oh boy, we're getting into all kinds of stuff. All right, here it is. So watch what happens to temperature over the next 100 years. So this is what the finest maps, or the finest models of changing temperatures out into the next 100 years show about temperature change in North America. So imagine how that's going to impact the distribution of Aedes aegypti and the other mosquito that transmits Aedes albopictus. So when you think about emerging diseases in Wyoming, keep in mind that what we have today and the conditions under which we exist could very well change off into the future. It could be a very different world. Let's see if we can get back to my okay, that was scary. It was worse than driving up on those icy roads today, Paul. Okay. So what we've been talking about is emerging diseases. So these are like first-time colonizations of human. We also have re-emerging diseases. So these are things that periodically pop into human populations, cause disease, and then they kind of disappear for a while, and then they come back. Okay? So these are diseases that once were major health problems that decline dramatically but are again becoming health problems. Some examples, malaria, something called cryptosporidiosis, which you get by drinking contaminated water, whooping cough, schistosomiasis, which is a worm that infects, where did my water go? Okay. I don't know whose bottle that is. Okay, schistosomiasis, which is a worm that lives in the bloodstream of humans, and measles. And some of you might be aware that one of the reasons measles and pertussis is coming back is because people are not vaccinating for these diseases in some cases. Other things that can impact or that can allow these things to reemerge are changes in environment. Schistosomiasis is coming back because of water development projects which provide suitable habitat for snails, which are the reservoir for schistosomiasis, coming back in many regions of Africa, Middle East, and South America. Changes in behavior, changes in resistance in the host population, and migration and dispersal. This just shows here this migration and dispersal. This is one of the ways that influenza virus spreads around the globe. And this just shows in the pink are the West Pacific Flyway. And this is where waterfowl actually moves up Southeast Asia, across Asia, where most of our influenza viruses originate because of agricultural practices there, gets into birds, and they carry it over across into Alaska, and it can spread out of Alaska down into North America. A really interesting case of a re-emerging disease is what's going on in Venezuela right now with malaria. And for those of you, am I okay? Oh, I thought you were going to kick me. You were bringing the hook. You're way over time, buddy. Thank you, Paul. So malaria, if you're not aware, malaria is all the blue lines on the right, that's your right, that shows the life cycle of malaria in humans. We get it from the bite of a mosquito. The malaria organism, what's called the trophozoite, colonizes red blood cells. It lives in the red blood cells, it multiplies in the red blood cells, and then the red blood cell explodes and releases little malaria parasites out into the blood, and they infect more red blood cells. This goes on and on. The individual that's afflicted goes through these periods of what we call paroxysms, very high fever, chills, shaking, muscle aches, curled up in the corner, and then they clear all of the damaged red blood cells, and they get up and they walk around and they feel great, and then this thing does it again in these cycles of red blood cell infection and eruption. At a certain point, malaria produces stages that are actually the sexual stages. They're called the gametocytes. And when a mosquito feeds on blood, it picks up those gametocytes, and now we get over on the red side. And malaria develops in the mosquito. And then the malaria organism lives in the body cavity, it multiplies, and the little infectious malaria trophozoites crawl up and infect the saliva glands, salivary glands. So when the mosquito bites, and its saliva, which is actually an anticoagulant, so it sticks its little proboscis in you like a hypodermic needle, it spits in you this anticoagulant saliva, and there are the little malaria parasites there. Quick biology of malaria. Okay, in Venezuela, malaria was virtually eradicated in 1961. It was gone. It was not a concern. They had mosquito control. Venezuela was feeling very good. In 2016, there were almost 250,000 cases of malaria. 2017, there was a 68% increase over 16. So what's going on in Venezuela? You read the papers. There's a lot of civil unrest. The economy is in the ditch, hyperinflation, economic and medical infrastructure collapse. What's happened is in the big cities, people, especially men, bankers, teachers, on the weekends and on their vacations, they're moving out of the city into the country, and they're engaging in illegal gold mining to make money, to get by. They turn around and they come back into the city and they don't feel well. So they're bringing malaria, which is becoming increasingly prevalent out in the countryside, and they're bringing it back into the city. It's spreading all over Venezuela, and now it's starting to spread across that region of South America. Just a photograph here. The red line shows the increase in malaria in Venezuela so far, and this graph goes through 2015. And the picture at the bottom just shows a camp of gold diggers, gold miners. And you can see part of the gold washing process creates these lovely, filthy ponds, which are ideal for mosquito breeding and larval development. Okay, a couple other things I want to mention just in the way of emerging diseases is there are some things that seem to be emerging, but it's probably because we have developed better technology and we're now becoming aware of them. And one of them is something called Toxoplasma Gondii. And this is a parasite that actually you can thank your house cat for this one. We'll talk a little bit more about this in a few minutes. But Toxoplasma Gondii is now easily identified and is now recognized as one of the top 3 sources of food poisoning, food poisoning in the world. Naegleria fowleri causes a very tragic disease called primary amoebic meningoencephalitis, which you get from swimming in reservoirs and sucking the water into your sinuses. Don't do that. And then there's histoplasmosis, which is something that we get from bats, and having bats in the attic, especially in your old cabin, I would encourage you to put screens up and keep them out. Okay, so just very quickly, because I'm running out of time and I know it's getting late, the environmental factors in the emergence of infectious diseases that are pretty widely known to facilitate these types of things. Ecological changes, things that change the relationships of humans and those reservoir animals out there. Just give you an example, building a housing development out in an old prairie dog town is a little bit of a risky endeavor, or next to a prairie dog town. A plague is still here. People still get plague, and that just puts people in proximity to prairie dogs, which is a risk. Human demographics and behaviors. I mentioned malaria in Venezuela. International travel is a way to disperse these things. Technology and industry. Now, one of the things we do in terms of food processing that facilitates outbreaks of disease is we've centralized a lot of things. And when you have an E. coli outbreak, which causes hemolytic uremic syndrome, well, the reason you have an outbreak is because all of that beef went through a processing plant and then got distributed widely in burger to a wide segment of the population. Of course, other things that you're all aware of are things like microbial antibiotic resistance. And then another thing to think about, and something that I worry about in these days when I read about budget cuts to health agencies, is our public health infrastructure. Okay, let's look at Wyoming. Wyoming's pretty good. This is a map. It's actually produced. You can go look at it on the web. This is by a program called PREDICT. Okay, PREDICT was created in 2009 to improve global detection and discovery of zoonotic viruses and with pandemic potential. Things like SARS and MERS and Nipah, Hanta, Ebola. Okay, and this is the combined effort of a lot of different national and international agencies to create this, and it's actually run out of the University of California at Davis. Just want to mention in the corner there, part of their program is about training people. So they've trained over 3,000 people around the world on how to sample animals, how to test them for different viruses, how to sample people. There are 60 PREDICT labs around the world. They've sampled 74,000 people and identified 820 new viruses, never known before. This is since 2009. And they found in new areas 182 known viruses. Okay, some things of interest to Wyoming and to the Rocky Mountain region. One is the Hantavirus respiratory syndrome. How many have heard about Hanta? Okay. Hanta in 1993, there was an outbreak in the southwest United States in the Four Corners region. They discovered a new variant of Hanta, which they named the sin nombre Hantavirus, which means the no-name Hantavirus. Mortality with this virus is about 38%, so it's not something you want to mess with. Early on in the outbreak, they determined that the reservoir for Hanta were deer mice. And what they found out was deer mice, especially during years of high pinion nut productivity, do very well. And they really move into houses and into storage sheds. And when people go in to clean these things up, what happens is you're sweeping. You probably all know this. This is pretty well known in Wyoming. People are careful. But you go in to sweep and clean up, and you raise a lot of dust up. You aerosolize the virus, you breathe it in, and this is how you get Hanta, is in these enclosed spaces where you stir it up. And there have been a lot of deer mice. Prevalence in deer mice runs about 10%, so it's not real common in deer mice, but if you get enough of them in these high deer mice productive years. One interesting observation, there are a lot of people, field biologists, that work with deer mice, and this is actually me and my crew up in Yellowstone, and we've got a deer mouse in the bag. People that have worked with deer mice for 50, 60 years, handled tens of thousands of them. They've never, you can get Hanta and get sick and recover. You can measure if you can determine if a person has had Hanta by looking at their, what's called their antibody titer. No mammologist that works with Hanta has ever been found to have an antibody titer to Hantavirus. So it really requires those closed spaces, lots of mice defecating, urinating, and then stirring it up, so it means it's pretty hard to transmit, actually. So just looking at the cumulative case count of Hantavirus across the region, and you can see in Wyoming there have been 16 cases. So Hanta is something to be aware of, but its potential to be a serious emerging disease is limited because of the difficulty of transmission. And it's very easy to cut off just by practicing the simple recommendations that public health officials recommend. Damping down the room, using a dilute bleach spray, which immediately kills the virus. And this just shows the potential for Hanta to occur across North America. This is the distribution of deer mice across North America. Okay, West Nile virus. West Nile was first identified in 1937 in Uganda, first arrived in the United States in Queens, New York in 1999. Nobody knows how it got here, but it's suspected that it might have been a stowaway mosquito on a jetliner or a bird or something. It was first recorded in Wyoming in 2002. And as you can see, the reservoir for Hanta are birds, particularly corvids, so ravens, magpies, blackbirds. Mosquitoes bite the birds, pick up the mosquito, can infect humans, and also horses. So this is a problem for horses. One of the amazing things about West Nile is how quickly it's spread across the country because birds are so mobile. And the mosquitoes, there are a number of mosquitoes that can transmit it. So here's the original occurrence in 1999 in Queens, New York. Okay, here we are in 2001. Here we are in 2003, and here we are today. In 15 years, it crossed the entire country. Here we are in Wyoming, and you can see, in particular, up in this area, there have been horses diagnosed. The light green is animal cases. The dark green are human cases. And in Wyoming, the greatest prevalence, highest occurrence of West Nile virus are in the areas with a lot of agriculture, which means there's a lot of water development, which allows for mosquito breeding. And I'll just mention, going back to our work with my NIH program, this is Eric Atkinson and a student. They're up at Northwest College, and they're doing work on Hantavirus and other diseases in birds, and this is funded through our NIH program. And that crazy-looking kid there at CWC is Tyler Graham, and at Central Wyoming College, they're looking at Hantavirus and mosquitoes, working with public health officials. Tyler's now in optometry school in Nebraska. So what about Wyoming? If we think about those environmental characteristics that facilitate emergence of disease, we, this just West Nile virus and mosquito habitat, when we talk about ecological changes, Hantavirus and deer mouse population densities, which affect the prevalence of Hanta, again, ecological changes. A number of things as we increase, as people move from rural areas into cities. International travel, of course, we have a lot of international travelers in this state with the energy industry. and the potential to bring influenza in and other things like that. Technology and industry. One thing, and I'm going to talk about this in a minute, and I'll talk about it really quickly, is an interesting disease we have in Wyoming that people worry about an outbreak, but it's probably not something to worry so much about, and that's chronic wasting disease, which is of concern to people that hunt deer, elk, and moose. We, of course, have microbial adaptation issues. I teach a lot of students that work in health clinics in Casper, and they talk a lot about MRSA and MRSA testing in the office. Okay? So how am I doing? 2 minutes. Okay, I want to say something about chronic wasting disease. Is that okay? Okay. So chronic wasting disease is a transmissible spongiform encephalopathy, what we call a TSE. Okay, this is the same thing that mad cow disease, which several years ago in the United Kingdom was a big concern, led to what's called non-variant Jakob-Creutzfeldt disease. A number of people fairly young developed a dementia-like disease, and it was traced back to the beef being produced in the country and the way they produce beef and the things they feed to cows. And this bovine form was able to switch hosts and move into human hosts with tragic consequences. We have a similar thing in North America. It's a transmissible spongiform encephalopathy. It affects deer, whitetail, mule deer. It hasn't been diagnosed in moose in Wyoming, but also elk. It has not been demonstrated to cross into humans, so it hasn't switched hosts that anybody knows about. It has been in the lab transmitted to primates. They're pretty closely related, so it is something to think about and to pay attention to those guidelines for how to handle your animal every fall. It is very prevalent in Wyoming, in our deer. In some regions in Casper, around Casper, 28% of the deer have tested positive for CWD. These are just some numbers from Wyoming Game and Fish. You can see that they range from 8 to 20%, depending on, those higher numbers are actually in targeted samples. So this is where they see an animal behaving oddly, and so they take it just to see if it does have CWD. Okay. One last disease. One last disease. I just want to talk about one other silent emerging disease. This is one that we don't, we're just discovering how much of it is out there. We never realized, and it's because of the technology and our ability to uncover such things. And this is one called Toxoplasma Gondii. This is actually in my book. That's why I wanted to talk about it. Okay. As I mentioned, this is a disease. This is the leading cause, or depending on who you talk to, one of the top 3 leading causes of foodborne illness-related death and hospitalization in the US. Now, people have known about this organism for quite a while, and those of you that have ever had children, it's now pretty common practice that when you go to the doctor and he says you're going to have a baby, and in the ensuing conversation they should be asking you if you have a cat. And if you have a cat, then they look to the non-pregnant partner and say, you take care of the cat box for the next so many months and keep your partner away from the cat box. And that's because of this organism. This is one of the most fascinating organisms in the world. It infects every warm-blooded animal. Birds, mammals, humans, and it is everywhere. Polar bears. Studies of 20% of polar bears test positive for toxoplasma. Grizzly bears. 25% of grizzly bears in Wyoming test positive for this. Walruses. 18% of walruses. Okay? It's bizarre. Humans, about a third of all humans test positive for this parasite. This parasite, it has two hosts in its life cycle. One's called the definitive host, and this is the one that produces, it's kind of the nexus of the life cycle, and the only definitive host are cats. Any type of felid, house cats in particular. These cats pass these little infective things called oocysts, and those oocysts will infect anything. Rodents, probably the natural host, so it's an old cat rodent life cycle, but now they get into everything, pigs, horses, sheep. So the source for humans is typically either from the cat box, from cat droppings out in the wild, or from eating undercooked meat that is infected with this toxoplasma. The interesting thing about toxoplasma, a couple of things. If it infects a pregnant woman during the first trimester, it can result in some very severe birth defects and developmental problems, much like Zika, mind you. So microcephaly, impaired cognitive development, and so on. When it gets into people that aren't pregnant, once you get it, you've got it. And what happens is it migrates out into the tissues and it insists in what are called hypnozoites. And it just sits there, and it's kind of like this, your immune system is controlling it, keeping it bottled up, and it just sits there in this sleeping state, we thought. Well, one of the tissues that it colonizes are nervous tissues. And it gets into the central nervous system. And there, in the last several years, have been all these crazy studies coming out that those hypnozoites aren't really asleep. They're actually producing chemicals and interacting with the nervous system, doing things like moderating dopamine levels and causing inflammation. And so these studies are coming out now where people that are infected with toxoplasma that test positive for it tend to have more aggressive behavior. A paper in Moscow, came out of Moscow, Russia a couple of weeks ago that I just stuck into our book. Car accidents. Significantly more car accidents involve people with toxoplasma than people that don't have toxoplasma. Studies at the University of Wyoming, funded by our NIH program. People with Huntington disease, which is a form of dementia that is genetically controlled. People with Huntington's and toxoplasma progress more rapidly to severe disease. People with Parkinson's progress more rapidly to the more severe stages of Parkinson's. It's crazy. So, but this is something we're just learning, something that's been out there, something that is emerging in terms of our knowledge. But here's just a bunch of, went around clipping out headlines. Toxoplasma, a common parasite that makes you angry. Can your cat cause mental illness? How worried should cat owners be? Cat parasite linked to cancer, Alzheimer's, and Parkinson's. Toxoplasma may be linked to crazy cat lady syndrome. When you go back to the mouse-cat deal, one of the really interesting things about mice is mice that are infected with toxoplasma, they have no fear of cat urine. So if that cyst wants to get back to a cat, what could be better than a mouse that walks up to the cat box for the cat to eat? So, in conclusion, this is kind of changing direction a little bit. Should Wyoming worry? I mean, there are things out there that can get you. But one of the things in putting together this talk that is incredibly impressive, and I have been Googling like I've never Googled in my life, looking at stories, looking at resources, and the infrastructure out there that is watching, that is sampling, that is monitoring, that is tracking is astounding. And it's saving us from a lot of these things that could infect us. The knowledge base is growing leaps and bounds. Just wait till our book comes out. It's just going to blow the roof off of this. The technology is improving every day. I mean, we're now at the point where, you can go out and for some diseases, we have diagnoses on a card. You can take a drop of blood, put it on something the size of a credit card, get a color change and say, bingo, you've got X. Let's put you in isolation or whatever it might require. This is just a table. I'll just leave it up there for you to look at while we address questions. It just shows some of those internet-based resources that are out there. Everything from social media. I love this, Foodborne Chicago. You can actually report when you're not feeling well after eating at that hot dog stand, so your friends won't go there. But listen, I want to thank you. I hope you heard some things that were interesting, and I'm more than happy to take any questions you might have. Yes?
Is there a treatment or a cure for toxoplasmosis?
There is not. There is not. I mean, what, these. Oh, I'm sorry. So the question was, is there a treatment for toxoplasma? And at this time, there is not. These are interesting findings, but there's a lot of debate whether these are robust studies. But there are enough of them, and some of the biochemistry of what people are learning about what those encysted parasites are doing biochemically in those cells, and the fact that associated with them is a little inflammation. And chronic inflammation is clearly something that can stimulate rapid cell division. Rapid cell division causes more mutation, which can result in cancers. Yeah, but there is no treatment for toxoplasma.
So do all cats have it?
Do all cats have it? My understanding, and perhaps one of the great things about being in Wyoming, is it's fairly rare in cats in Wyoming because this is such a dry environment. So I have heard that. I looked high and low for a survey of cats in Wyoming, domestic cats, and I did not find, nobody's done it. Maybe we ought to do that. Yeah, Matt.
It's not like, be the odds maker for Las Vegas. What do you think the odds are that, say, in the next century we have a global pandemic, but something, akin to Ebola, Marburg, maybe that gets aerosolized and that leads to profound human tragedy? Our population's so big and all that. What are your thoughts on that?
So, I thought it'd be fun to show a little video of World War Z just for kicks, but I think that's copyright infringement. So, the last really significant epidemic was probably the early 1900s, the influenza H1N1. And that was tragic, and that about 8 to 10% mortality in the regions where it occurred. And that's a lot of people. I think with the technology, and especially, and I was going to talk a little bit about, I think tomorrow I have a few more minutes, I'll talk a little bit about this new CRISPR-Cas9, where they're now talking about the ability to quickly manufacture very specific antimicrobials that can target viruses and bacteria. And that is quickly coming to development. And I think our technology, as long as we continue to invest, and I'll go back to a comment I made earlier, there's some fantastic resources out there, but they're not for free. And they're doing yeoman's work to protect us. And when I hear about budget cuts to the National Institute for Health and the Institute for Allergies and Infectious Diseases, and World Health Organization suffering from countries cutting their contributions, that worries me, because there are people out there that are watching, alerting, and I think preventing the opportunity for those types of things. But so I can't guess, and I'd be recorded, and then I'd be.
So let me take the moderator's privilege and ask one last question, which I hope you can give me a good answer to. Should
Yes.
we all go home? Should we seal our doors and windows? Should we wear face masks? Where are we?
No, you know, like I said, I think that, you know, in particular North America and the United States, we have wonderful health care. We have invested a lot of resources that have been used very wisely. We certainly don't want to back away from that. And I think pay attention to what public health people tell you to do. Things like there is no evidence that vaccines are related to the things that are claimed. Absolutely none, not a single paper. So get your kids vaccinated. It's very important.
Thank you very much.
Okay. Thank you.